Good day, this is Professor Will, CBMD, PhD. Today's Tuesday, November 23rd, 2021. It is 7:06 and 30 seconds, p.m. Eastern Standard Time. Today's research, ultra-short pulse lasers. So let's hop to it here. Ultra-short pulse lasers kill bacterial superbugs, spores. Technique likely safe for human cells. Has potential for sterilizing wounds. Blood products. Date November 23rd, 2021. Source: Washington University, School of Medicine. The researchers previously have shown that such lasers don't damage human cells, making it possible to envision using the lasers to sterilize wounds or disinfect blood products. The ultra-short pulse laser technology uniquely inactivates pathogens while preserving human proteins and cells. Imagine if, prior to closing a surgical wound, we could scan a laser beam across the site and further reduce the chances of infection. The researchers can see this technology being used soon to disinfect biological products in vitro and even to treat bloodstream infections in the future by putting patients on dialysis and passing the blood through a laser treatment device. Researchers have been exploring the germicidal properties of ultra-short pulse lasers for years. They have shown that such lasers can inactivate viruses and ordinary bacteria without harming human cells. The researchers trained their lasers on multi-drug resistant Staphylococcus aureus, which causes infections of the skin, lungs, and other organs, an extended spectrum beta-lactamase, producing Escherichia coli, which causes urinary tract infections, diarrhea, and wound infections. Apart from their shared ability to make people miserable, MRSA and E. coli are very different types of bacteria, representing two distinct branches of the bacterial kingdom. The researchers also looked at spores of bacterium Bacillus cereus, which causes food poisoning and food spoilage. Bacillus spores can withstand boiling and cooking. In all cases, the lasers killed more than 99.9% of the targeted organisms, reducing their numbers by more than 1,000 times. Viruses and bacteria contain densely packed protein structures that can be excited by an ultra-short pulse laser. The laser kills by causing these protein structures to vibrate until some of their molecular bonds break. The broken ends quickly reattach to whatever they can find, which in many cases is not what they had been attached to before. The result is a mess of incorrect linkages inside and between proteins, and that mess causes normal protein function in microorganisms to grind to a halt. At a certain laser power, we are inactivating viruses. As you increase the power, you start inactivating bacteria, but it takes even higher power than that, and we are talking orders of magnitude to start killing human cells. And so there is a therapeutic window where researchers can tune the laser parameters such that they can kill pathogens without affecting the human cells. Heat, radiations, and chemicals, such as bleach, are effective at sterilizing objects, but most are too damaging to be used on people or biological products. By inactivating all kinds of bacteria and viruses without damaging cells, ultra-short pulse lasers could provide a new approach to making blood products and other biological products safer. Anything derived from human or animal sources could be contaminated with pathogens. Researchers screen all blood products before transfusing them to patients. The problem is that researchers have to know what they are screening for. If a new blood-borne virus emerges, like HIV did in the 70s and 80s, it could get into the blood supply before they know it. Ultra-short pulse lasers can be a way to make sure that our blood supply is clear of pathogens, both known and unknown. Good day, this is Professor Will, CBMD, PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.